Neutrophils play an important role in chronic inflammatory conditions and acute viral and bacterial infections in the lung. In order to study the mechanisms of neutrophil trans-epithelial migration into the airways of children with severe RSV bronchiolitis, we have developed a method using ciliated airway epithelial cells grown from nasal brush biopsies.
Our gravity-fed system supports neutrophil migration in a physiologically relevant basolateral to apical direction. This technique allows us not only to measure the numbers of neutrophils that migrate through, but also to observe their movement and the impact of neutrophil migration on epithelial cell ciliary beat frequency under the microscope.
It is important to coat the membrane inserts on both sides with collagen to allow adhesion of the epithelial cells. This procedure should be performed in a sterile environment using a Class II safety cabinet. Add 100 microliters of collagen working solution to the top of the membrane inserts. Next, replace the lid and invert the plate. Set it down and remove the plate bottom, leaving the membrane insert standing upright on the plate lid. The collagen solution below the membrane insert remains in place by surface tensions. Ensure the solution does not drip down the side of the membrane insert. Now very gently add 100 microliters of collagen working solution to the other side of the membrane insert. Replace the bottom of the 24-well plate to cover the membrane inserts, being careful not to disturb the collagen solution. Leave at room temperature for at least 1 hour. After incubation, aspirate the collagen solution from both sides of the membrane insert. Next, invert the plate to remove the collagen from the other side of the membrane insert. Rinse each membrane insert with sterile water on both sides of the membrane. Leave the lid off the plate and allow the collagen-coated membrane inserts to dry in the tissue culture hood for 1 hour. Once the membrane inserts have dried, replace the lid and seal the plates using parafilm. These plates can be stored at room temperature for up to 1 month. To prepare the membrane inserts for seeding epithelial cells, invert the inserts in a 12-well plate using tweezers. Prepare your epithelial cell solution to 1 × 10⁶ epithelial cells in 70 microliters of F media. Add 70 microliters of cell solution to the inverted membrane inserts, being careful not to leave any bubbles in the seeding solution. Replace the lid and incubate at 37 degrees with 5% CO2 in a humidified incubator for 4 to 6 hours. Then, remove the plates from the incubator and return the inverted inserts into a 24-well plate. Add 500 microliters of fresh F media underneath the membrane inserts. Add 100 microliters of F media, supplemented with 30 micrograms per ml collagen and 5% matrigel to the top of the membrane insert. Incubate the seeded membrane inserts for 24 to 48 hours at 37 degrees 5% CO2. After 24 hours, aspirate the media from both sides of the membrane insert. Add 100 microliters of Ali media to the top of each membrane insert. The cells are now at air-liquid interface. Incubate the membrane inserts at 37 degrees 5% CO2 in a high humidity incubator and feed the cells every 1 to 2 days for up to 28 days to allow cellular differentiation. To view motile cilia, use an inverted microscope equipped with a long working distance ×20 objective. Our images were acquired at a resolution of 0.22 micrometers per pixel. Prior to infection, rinse the apical side of the cells with 400 microliters of alley media to remove any detached epithelial cells. Then aspirate the media from both the apical and basolateral sides of the membrane inserts. To infect the epithelium, gently remove the membrane inserts from the 24-well plate and place upside down in a fresh 12-well plate. Pipette 25 microliters of virus preparation onto the cell side of each membrane insert, being careful not to touch the airway epithelial cells. Place 25 microliters of control media onto the mock-infected epithelium. After 1 hour incubation in the incubator, aspirate the infection media and wash the cells with PBS to remove any excess liquid on top of the membrane inserts. Place the membrane inserts into a fresh 24-well plate and add 100 microliters of alley media to the basolateral side of the cells. The apical side of the membrane insert remains at air-liquid interface. Incubate at 37 degrees 5% CO2 for 24 to 72 hours to allow the progression of viral infection. On the morning of the neutrophil migration day, place 600 microliters of alley media under the membrane inserts for 30 minutes to allow collection of chemokines and cytokines produced from the RSV or mock-infected airway epithelial cells. Centrifuge at 1000 G for 3 minutes and discard the pellet containing any detached epithelial cells. During the spin, transfer the membrane inserts into new ultra-low binding 24-well plates. Neutrophils do not adhere to the surface of these plates, so can be collected post-transmigration. Replace 400 microliters of the supernatant underneath the corresponding test condition membrane insert. Aliquot and store any remaining supernatant at -20 degrees for future analysis. As a positive control, add 400 microliters of 100 nanomolar FMLP to the underside of a mock-infected membrane insert. Return the plate to the incubator until the neutrophils are ready. Purify neutrophils from venous blood and stain with a viability stain such as calcine red-orange. Those neutrophils which pass through the transwell inserts will then be collected and counted. On the bench, prepare a neutrophil solution at 5 × 10⁶ neutrophils per ml, supplemented with 1% autologous serum. Then add 100 microliters of this solution to the basolateral side of each membrane insert. Incubate the plates at 37 degrees 5% CO2 for 1 to 4 hours. Different time points can be used depending on the experiment. After migration, collect the neutrophils from the basolateral side of the epithelial cells. These are now referred to as the non-migrated neutrophils. Then, collect the neutrophils on the apical side of the epithelial cells. These are now referred to as the migrated neutrophils. If the neutrophils were pre-stained, at this point you can quantify the number of migrated neutrophils using a plate reader and compare to a standard curve of known numbers of stained neutrophils, as described previously by our group. Finally, fix the membrane insert in 1% paraformaldehyde and stain for neutrophil or epithelial cell markers of interest. Wrap the plate in Nesco film and store in the fridge at 4 degrees. These plates will be stable for up to 1 month.
Our results show that airway epithelial cells need to be grown on membrane inserts with a pore density no greater than 0.6 times 10 to the 6 per centimetre squared. Larger pore densities prevent the airway epithelial cells from being visualised under the microscope. When we compare the quality of cultures grown on the underside of a 3 micrometre pore membrane insert to cultures grown on the top of a 0.4 micrometre pore membrane insert, we found there was no significant difference in TEER at 28 days post-air liquid interface. We also found no difference between the average ciliary beak frequency and number of ciliated cells present on these cultures.
Although this method was developed for exploring neutrophil migration through airway epithelial cells in RSV bronchiolitis patients, it can be modified to analyse neutrophil function in other types of respiratory infections and other inflammatory diseases, using cultures grown from biopsy samples obtained from patients with COPD, asthma, cystic fibrosis, for example.